Good day to everybody. Today, I am my topic of discussion is DNA, deoxyribonucleic acid. DNA. In my previous video, I have discussed about the chromosome, and I have told that this DNA is present. DNA molecule is present inside the chromosome. Now you can think about one aspect of this situation like that. When any truck or any tanker carrying any commodity commodity or goods like milk, any tanker is carrying any milk, then we are concerning about the milk mainly, not we are concerning directly about the tanker. Like that, chromosome is carrying the gene or DNA, deoxyribonucleic acid, but chromosome has its own importance in heredity or inheritance, but it has not directly impact the our characteristic and traits, which is the actually DNA is responsible for that, and we are mainly targeting the DNA. We are thinking about the DNA, and we are concerning about the DNA, deoxyribonucleic acid. So this is the history of DNA. James Watson and Francis Crick, these two scientists, in 1953 published an article entitled "A Structure for Deoxyribonucleic Acid," and they have received Nobel Prize in 1962 for this work. And actually there this discovery actually opens the door of several new emerging fields like recombinant DNA technology, genetic engineering, biotechnology, etc. Now come to the point, what is DNA and what is the structure of DNA. Actually, they have described, the Watson and Crick have described the DNA structure as a double helix structure. Double helix means twisted ladder or twisted staircase like structure. Now I am showing you by drawing some diagram, so how it is looking like the double helix structure of deoxyribonucleic acid DNA. Actually, it is looking like that. This is like almost like that DNA. Double helix structure. I have told you this is double helix structure. And this is the two strands of DNA. And these strands are complementary to each other. This is one strand and this is one strand. And they are complementary to each other, and they are also anti-parallel to each other. Means in opposite direction. One strand in opposite direction to the another strand. Now coming to the point of DNA, what is actually DNA made of? The DNA, the three components, main components of DNA, deoxyribonucleic acid, is first one is heterocyclic 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 nitrogenous ring nitrogenous ring or base, which may be purine or pyrimidine. Two types of this nitrogenous base will be present in DNA. And second one is deoxyribose. deoxyribose. It is a 5 carbon sugar which is also called pentose, and it is also deoxyribose it is called. And third one is the phosphoric acid. Phosphoric acid. Now I will gradually discuss about showing the structure of these three components so that you can understand it better. Now first we come to the structure of uh, ribose, so deoxyribose, which is present in DNA, deoxyribonucleic acid, deoxyribose. How it is looking like? The structure of deoxyribose is like that. CH2OH, this is H, this is HOH, this is HH, and this is HOH. This is the structure of deoxyribose. Where I am not writing any letter, means the corner of the ring. You can imagine that this was the actually there are C carbon atom is there in at every corner. So this is the ribose structure, and this is the oxygen atom present. This is the structure of deoxyribose. And another in comparison with deoxyribose, another structure is the ribose structure. You can see the difference of the two deoxyribose structure and ribose structure. CH2OH, H, HOH, HOH, and HOH. This is the structure of ribose. You can see from clearly see that here the oxygen is absent. Here oxygen is present. So this is the reason it is called ribose, and this is called deoxyribose. This is the main difference between the structure of deoxyribose and ribose. Deoxyribose is present in the DNA molecule, and ribose structure will be present in the RNA molecule, means ribonucleic acid. Ribonucleic acid were present actually messenger RNA, ribonucleic acid, transfer RNA, ribosomal RNA. These are the RNA component present in the cell. So these are made of this ribose backbone structure. And this is the basic structure of the deoxyribose sugar and ribose sugar. And what is the difference between this is the main difference between the RNA and DNA structure. Now we showing the phosphoric acid structure, how it is looking like. This is the structure of phosphoric acid H3PO4. It is the another component of DNA, deoxyribonucleic acid. I have already discussed, and I will show you the two structure of purine and pyrimidine bases. Actually, this purine base of DNA will be two types. One is adenine, which is denoted by capital A. Another is guanine, which is denoted by capital G. And pyrimidine base, another base, nitrogenous base, heterocyclic base is pyrimidine, which is the there are three types of pyrimidine base. One is thymine, second one is cytosine, and third one is uracil. Actually, in DNA structure, adenine, guanine, thymine, and cytosines are present, both purine and pyrimidine base. But in case of RNA, uracil will be present in place of thymine. In case of RNA structure, this is the another difference between the RNA and DNA structure. One is the ribose and deoxyribose sugar, and another is the uracil pyrimidine base will be present in place of thymine in case of RNA, and DNA will contain thymine base. So now I will, for example, I will show one purine based structure and one pyrimidine based structure, so you will understand it better. Like one purine base means adenine structure will be like that. Here is N, here is N, and where I am now not writing any letter, it is indicates that it will be carbon atom present there, and so here is a carbon atom present there, and here is the, and here H, now carbon, here carbon atom also present, like that, and here is the, and here is the H, here is the H. It is the actually the structure of purine base adenine. It is the structure of purine base adenine. So you can see from this heterocyclic ring in which in nitrogen atom and carbon atom both are present inside the ring, and like that adenine structure. Another pyrimidine base, I will show you the structure of one pyrimidine base like that. It is like that. This is N, this is N, and this is H, this is H, and this is CH3. This is the structure of pyrimidine base thymine. So you can see the, you can identify the structure of purine and pyrimidine base. I have shown one adenine purine base and another thymine pyrimidine base. Now DNA, how the DNA double helix, mean two strands are present, both are complementary to each other, I mean both are completes each other, and they are opposite in direction, anti-parallel. Now how they are showing like I will show you by diagram, one diagram. It is the one strand structure of ribose backbone structure. Now here is the phosphate group will be present, and this is the 3 prime, and this is the 5 prime. This is like that. It will be 3 prime. Oh sorry, this is the 3 prime. 3 prime is here. 3 prime is here, and 3 prime is here. And like that, it will be connected with the ribose sugar, like that, like that, like that. Sorry, like that. This is the 5 prime. It is the 5 prime, it is the 5 prime, and it is the open phosphate group, and this is the OH, means the hydroxyl group is present there. It is one strand picture. Another strand will be looking like that. Another strand looking like that. It is 3 prime. It is the 1 2 sorry, this is the actually 5 prime, not 3 prime, 5 prime, 5 prime, and like that here it is 3 prime, like one is 5 prime, 3 prime, like that, and another is like that, which will be 3 prime, and this is the 5 prime. So you can see that here 5 prime is open in one strand, and here is the 3 prime, 3 prime is open in one strand. This is OH group. Now this will be like 3 to 5 prime, 3 to 5 prime direction, and this strand will be in the opposite direction, like 5 3 to 5 prime in opposite direction. You can see 3 to 5 prime and 5 to 3 prime. So they are opposite to each other, anti-parallel. Now here the adenine will be combined with thymine base, pyrimidine base. Purine base will be combined with the pyrimidine base by one double bond, double hydrogen bond, like that. Like G, guanine will be combined with cytosine by 3 hydrogen bonds, like that. T, thymine will be combined with by double hydrogen bond with adenine, and it will be like that. So adenine always combined with thymine with 2 weak hydrogen bonds. Guanine will always combined with the cytosine with 3 weak hydrogen bonds. This is the general structure of DNA, double helix structure of DNA. Now you know that 4 purine and pyrimidine base are present in DNA. So deoxyribonucleic acid structure. So there will be some ratio of like that, A adenine plus T. This is called molar ratio, divided by G guanine plus cytosine. This is called molar ratio. Molar ratio. And this molar ratio will be constant for different, for particular organism or species. And it is the main characteristic of any organism. You can identify the organism by this molar ratio. It is called molar ratio. And in human beings, this molar ratio is 1.52. For sheep, this molar ratio is 1.36. Like wheat germ, this molar ratio is 1.19. Like microorganism Escherichia coli, this molar ratio is 0.93. And in case of Clostridium perfringens and anaerobic bacteria, the molar ratio will be 2.70, like that. So for every microorganisms or every species and organism, this molar ratio are constant. It will be not affected by age, growth or environmental condition. Now come to the point. Another point is that there are 2 strands are present here, like A, T, double strand in DNA, C, G, C, C, T, A, G, like that. One strand will be like that, and another strand will be just complementary to each other. It will be T, A, G, C, G, G, T, A, T, G, C, like that. Another strand, it is 5 prime to 3 prime, and it will be 3 prime to 5 prime. Now what happened in for any protein synthesis, these strands should be separated by each other to for reading the nucleotide sequence present here. Here every three nucleotide sequence like adenine, thymine, cytosine, guanine, cytosine will be denoted by actually they are the codon or triplet. They are called codon or triplet. These three character actually specifies and represent one amino acid. You know that amino acid, and this is called codon. Three triplet, three nucleotide sequence will be called triplet and codon. We have actually this for protein synthesis. This double strand structure will be separated by some means like heat, and then another it will be read to produce the mRNA, messenger RNA for protein synthesis, which is called translation. I will discuss it later. Translation, mRNA will be produced from this strand, and which strand will be read to produce the mRNA is called sense strand. This is strand from one protein, and this is the sense strand for another protein. They will be separated and they will guide to produce the mRNA, messenger RNA, to produce the protein for our, and actually they are signifies, they are represent and specifies the amino acid, and then amino acids are combined to with each other to produce the protein by peptide bond, you know. So actually what will be the mRNA for this strand? I will discuss this strand. T will be it will be also a complementary strand will be T will be A will T will be A will be U. I have told that uracil come here. Not in case of structure of mRNA, uracil will come from this strand, from translation. G will be C, C will be G, G will be C, G will be C, A will be again U in place of T, because RNA structure will be like that, and T will be A and C will be G, no problem of that. Only in case of T, it will be replaced by uracil pyrimidine base, and these are called actually codon, not DNA. The DNA, the triplet structure will be called codon. Actually, in mRNA, this is called codon, and this will specify and represent one particular amino acid. Now there are four letters A, U, G, C. So we can get combination of this arrangement by three combination of this four letter arrangement. We will get 4 to the 3 is equal to 64 combinations of this letter. So, but we should get 64 combination. So it will represent 64 amino acids. But it is not happen because there are three terminating codons. So it is only 61 codons. There are only 61 codons. What are the terminating codons? I will discuss about that. And actually, in nature there are, we know that 20 amino acids are present. Among them, 10 amino acids are essential amino acids. Essential amino acids are those amino acids which cannot be produced by our body, synthesized by our body. We have to acquire it from the outer source, food source. So these are called the essential amino acids like phenylalanine, methionine, valine, isoleucine, leucine, lysine, histidine, arginine, tryptophan. They are all 10 amino acids are essential amino acids which our body cannot produce or synthesize. We have to acquire it from the outer source. So these three terminating codon, any chain of the amino acid chain, there should be some terminating codon where we have to stop the gene reading, or when the gene reading it will be identify these three codon, three letter, and these are the terminating codon. I will show you the which are the terminating codon, you can understand it better. Terminating codon are first one is UAA, which is called Ochri, UAG, which is called amber, it will not specify any amino acid, and UGA, which is called umber. These are the terminating codon. They will not specify any amino acid. They are actually terminating the indicates the end or termination of any gene structure. So these are the, and you have seen that 61 combination of codon is there, and only we have 20 amino acid. Means one amino acid will be specified represented by more than one codon. This is the main thing. Means one or more than one codon will specify or represent one amino acids, because we have only 20 amino acids in nature, but our combination will be, codon combination will be 61, arrangement will be 61. So 61 will identify 20 amino acid in total. Now there, the like terminating codon, there should be some initiation codon. What is this initiation codon? Initiation codon is like that. Initiation codon, which will actually indicates the starting of the any gene structure, which is called initiation codon. They are like two are initiation codon. One is AUG, which will specify or represent methionine amino acid, and another is GUG, which will specifies valine amino acid. So from with methionine and valine, we can start the gene structure for protein production. So this is the all about the DNA structure and RNA structure and the mRNA, how we can read. And generally the mRNA will be made by 5' to 3' direction. In every case, in case of translation, this is called translation when we are making mRNA from DNA replicate. The DNA replicate we are making it, it is called translation. So this is all about the DNA and RNA structure and how the mRNA will be made, and the codon, what is codon, what does it mean. So you understand, I hope that you understood it very well, and if you like my video, please share and subscribe it. Thank you.